Hi, my name is Bob Wong. Thanks for joining the webinar. I'm a researcher based in Melbourne, Australia, which is situated on the traditional lands of the Boonwurrung and Woiwurrung people of the Kulin Nation. And I want to take the opportunity to acknowledge their deep cultural connection to country and also to pay my respects to elders past, present, and emerging. So I'm a behavioral and evolutionary ecologist, and for the last 15 to 20 years, I've become increasingly interested in trying to understand how human-induced changes in the environment might disturb the behavior, ecology, and evolution of wildlife. And I'm going to be talking about some of our research today. But I'm actually recording this from quite a cold day here in wintry Melbourne, so I do want to take the opportunity to temporarily transport you to somewhere a bit more exotic, the Sierra Madre up in Mexico. Which is where I spent some time as a postdoc in the United States when I was there straight after my PhD. And I'm going to share with you a story about a group of fish known as swordtails, so called because males of some species possess a tail-like extension, a sword-like extension of the tail fin, which gives this group their common name. So in this particular image that you can see here, there are actually two different species of swordtails interacting with each other. The fish on the bottom is a typical looking swordtail fish. You can see the tail ornament at the base of the tail here. It's a species called Xiphophorus malinche, and it's interacting with a male of another species, Xiphophorus birchmanni. Now, in some rivers in Mexico, there's two species actually coexist, and one of these rivers is the Rio Carnali. Now, like many other rivers in Mexico and indeed in many other parts of the world, the Rio Carnali is facing increasing pressures from human activities. You can see that there is rubbish strewn along the banks of the river, and the water has been stained this very dark color as a result of an increase in nutrient input. Indeed, in the 1990s, the township of Carnali had installed a sewage system that pumped black water directly into the river. And around the same time, researchers noticed that the two species of swordtails that had previously coexisted side by side in this river had actually been replaced by hybrids. So here are pictures of some males of these hybrid swordtails, which bear, I guess, resemblance to males of both of the original species. So as an early career researcher, junior postdoc, members of the lab that I had joined were interested in trying to understand what could be driving these hybridization events. And we already know from previous work done on swordtail fish that females actually pay a lot of attention to the smell or the odor cues of males. And in fact, odor cues play a really important role in mate recognition and reproductive isolation between species. So that was our starting point. So we went down to Mexico, we collected the two species from other locations in Mexico, and then brought them back to a research facility close to the Rio Carnali, and we started to test the females and their preferences for the odor cues of males of their own species versus the odor cues of the related other species. So here we have some results. So these are results for Xiphophorus birchmanni females when they're offered the choice between the odor cues of males of their own species versus the odor cues of males of the closely related Xiphophorus malinche. And what we found was when we tested the females in clean spring water, females spend significantly more time associating with the odor cues of males of their own species. And the same was also true when we tested females in treated municipal tap water. However, when we tested females in the polluted water that was drawn directly from the Rio Carnali itself, in fact, water that came very close to the sewage outflow, we found that this preference for the odor cues of conspecific males disappeared. So the reason why I wanted to share this early piece of research with you today is just to sheet home the point that altered reproduction and indeed altered reproductive behaviors in this case can have important ecological and evolutionary consequences. So from an ecological perspective, we've seen the replacement of two species in this ecological community with a hybrid swarm. And from an evolutionary perspective, we see a breakdown in reproductive isolation mechanisms that kept two species that were previously isolated, bringing them together again, resulting in the loss of biodiversity and in fact changing the course of evolution itself. But let's return back to Australia. And a few years ago, we decided to look at how pollutants might also be affecting the behaviours of fish back here at Monash. And one of the early pieces of research that we did actually involved collecting fish from a couple of locations in urban Melbourne. So this is one of the study sites. It's located just downstream from a sewage treatment plant that was pumping treated effluent into the river itself. So we collected a species of fish known as eastern mosquito fish. So this is a species that belongs to the same family as the swordtails. So eastern mosquito fish, in this case, are native to North America, but they've been introduced to temperate and subtropical regions right around the world, including here in Australia. So we collected fish from two locations, one which was relatively pristine. It was adjacent to a reservoir that was collecting water for human consumption. And our second location was just downstream from the wastewater effluent discharge. And we brought them back into the lab and we looked at the reproductive behaviors of both the males and the females. I'm just going to quickly show you the results for the males. So again, we have a measure of association time. In this case, the amount of time that males spent associating with females, those males that were collected from pristine site versus those males that were collected downstream from wastewater effluent discharge. And again, we found that there was a shift or change in reproductive behaviors between the two locations. And in this case, the males from the wastewater treatment site actually showed an increase in mating activity, which is actually not a great outcome when you consider that eastern mosquito fish are one of the most invasive species in the world. So an increase in reproduction is not necessarily a good thing for the local ecological community. Now, both of these examples, I think, underscore the fact that chemical pollution is an insidious form of environmental change. And in particular, in recent years, concern has been mounting over a particular group of chemicals, the pharmaceuticals. Now, I'm sure everyone watching this webinar would appreciate the therapeutic benefits of the medicines that we take or that we give to our pets or to our livestock. But what's not very well appreciated is that vast quantities of these drugs can actually make their way into the environment. The reason for this is because many of the medicines that we take or their metabolites remain biologically active when they're excreted. And the fact is that most modern wastewater treatment processes, while very good at removing nutrients and solids, haven't been specifically designed for removing chemicals such as pharmaceuticals. So as a result, they do end up in the environment. And once they're in the environment, pharmaceuticals are a cause of ecological concern for several reasons. First of all, unfortunately, they become ubiquitous. So a paper published a few years ago, for instance, reported the presence of pharmaceuticals in the tissues of fish and invertebrates living adjacent to one of the research stations in Antarctica. So they're showing up even in some of the most remote regions on the planet. Another reason why they're cause for concern is because many of these drugs remain quite persistent in the environment. So one of the veterinary pharmaceuticals that we study, Trenbolone, has a half-life of some 270 days. Thirdly, because the receptors that are targeted by these drugs tend to be evolutionarily conserved, and what that means is that drugs that have been designed for you or I can also have effect on non-target wildlife. They can and do affect non-target species, often at very low concentrations. In some cases, we're talking about nanograms per litre. Now, an infamous example of the unintended consequence of pharmaceuticals in the environment was sheeted home by a study published a few years ago showing the population collapse of 3 different species of vultures on the Indian subcontinent as a result of birds ingesting livestock carcasses that had been tainted by a veterinary pharmaceutical. And of course, we also know that pharmaceuticals not only affect organisms in the terrestrial realm, but also in the aquatic environments as well. And one of the most infamous examples of this come from the effects of the contraceptive pill, which is showing up in waterways everywhere and has been linked to the feminization of male fish. Here in Melbourne, a study published a few years ago reported some 70 different pharmaceuticals in some of our urban river systems. And based on the amount of chemical contamination in stream invertebrates, the scientists estimate that platypus could be taking up as much as half a daily human dose of antidepressants every single day. Which brings me to fluoxetine, which is a drug of emerging environmental concern. Marketed as Prozac, fluoxetine is one of the most widely prescribed antidepressants in the world and belongs to a class of pharmaceuticals known as serotonin reuptake inhibitors, which, as the name suggests, acts by inhibiting the reuptake of the neurotransmitter serotonin, which I'm sure many of you would appreciate is involved in a whole range of important biological functions. And of course, fluoxetine has been designed specifically to induce physiological and behavioral effects in humans, for example. So we might expect it to also have an impact on wildlife. And like many other pharmaceuticals, is not completely removed during the wastewater treatment process. And as a result, it is ending up in the environment at concentrations ranging from less than 1 right up to 929 nanograms per litre. And we know that exposure of wildlife to fluoxetine can result in all kinds of sublethal effects. For example, it's been linked to impaired learning and cognition in cuttlefish. It's been shown to also disturb anti-predator responses in fish. And it's been found to affect foraging and activity levels in shore crabs as well. But what about reproduction? Well, we decided to investigate together with a then PhD student, Michael Bertram, who's now gone off and started a postdoc in Sweden. And so in particular, we were interested in looking at whether exposure of fish to fluoxetine might impact copulatory behavior of the males and also their sperm traits. And to do this, we returned to the eastern mosquito fish because their reproductive antics are very well studied because they are a model system in behavioral and evolutionary biology. So what's interesting about this particular species is that they are internally fertilized. So males have got a modified fin, which you can see in this image here, called a gonopodium, which he uses as an intermittent organ to inseminate the females. Now, this is a reproductive system that's dominated by male sexual coercion. So males are constantly trying to solicit copulations from females, indeed are constantly harassing females for matings. And prior research has shown that females suffer costs such as reduced foraging time as a result of this constant and incessant male harassment. So in terms of the exposure for this series of studies, we decided to expose fish to 30 days of fluoxetine. And the reason for this is because previous research has shown that at least in humans, the therapeutic benefits take around 3 to 4 weeks to manifest. And so we had 3 concentrations in this particular study: a freshwater control, of course, a low fluoxetine treatment, and a high fluoxetine treatment. You can see the measured concentrations reported in this slide here. And both the low and high concentrations that we use are reflective of levels that are actually found in the environment. So one of the things that Michael did was a series of behavioral trials, and this involved placing fish after the 30-day exposure into an aquarium, which you can see here, a single male with a single female, and over a 20-minute period recorded the number of copulation attempts performed by the male towards the female. In a separate set of studies involving different fish, we also stripped the sperm from the males, and we assayed a range of sperm traits based on established protocols. Now, the thing with mosquito fish sperm is that they're actually produced inside bundles that are delivered to the female. So these bundles had to be resuspended in activation solution. And thereafter, we looked at a whole range of things, for example, total sperm count by loading the sperm onto a hemocytometer and counting them using automated software. We also used computer-assisted sperm analysis, or CASA, to measure a range of sperm quality measures, namely various measures of velocity, path straightness, and sperm motility. And then lastly, or using commercially available test kits, we also looked at the percentage of live sperm as a measure of sperm viability. So how these test kits work is that they stain live sperm cells green and dead sperm cells red, and we're able to use software to count the percentage of live sperm. Okay, so turning now to the results. Does fluoxetine impact male copulatory behavior? The answer is yes. So here on the horizontal axis, we have the 3 different exposure treatments, the control, low, and high. And on the vertical axis, we have the number of copulation attempts. And what we found was that high fluoxetine exposed males had heightened copulatory behavior compared to the control. Well, what about sperm traits? Well, we found that fluoxetine exposure increased sperm count. So again, we have the different treatments on the horizontal axis, and we have a measure of sperm count on the vertical axis. And we found that irrespective of whether males were exposed to low or high fluoxetine concentrations, those males had significantly more sperm compared to the freshwater control. But we didn't find an effect of fluoxetine on any of our measures of sperm quality, nor did we find any effect of fluoxetine on sperm viability. So to sum up this part of the study, we found that exposure of male mosquito fish to environmentally realistic levels of fluoxetine affected male copulatory behaviors and also sperm number. Now, of course, it's important to bear in mind that the study that I just talked about only exposed animals to fluoxetine in isolation. Now, well, I guess most of you would appreciate that, of course, in the wild, animals are seldom confronted with just a single environmental stressor in isolation. More often than not, animals are exposed to a myriad of different stresses simultaneously. Things like temperature and also pathogens can be important stresses. So we wanted to see how those secondary stresses might interact with fluoxetine exposure to affect reproduction. So this time we carried out a series of studies with another PhD student, Lucinda Osselbrook, to look at the interactive effects of fluoxetine and other stresses on reproductive traits. And in particular, we're interested in looking at how fluoxetine might interact with pathogen exposure and also temperature. Our study organism in this case is a crustacean that's native to freshwater and brackish water habitats across Eurasia. It's something called a water flea, Daphnia magna. It exhibits an unusual mode of reproduction called cyclic parthenogenesis, and you can see some of the developing babies inside this individual here. So in terms of fluoxetine exposure for this particular study, the study itself was carried out over a 30-day period. We tested two different genotypes of Daphnia, M10 and HO2, because we're interested also to see if there might be some genotype-specific effects. And individuals were exposed chronically from 1 day of age. And in this case, we had 4 exposure treatments. We had a gain of freshwater control, a low fluoxetine, a medium fluoxetine, and this time around a high fluoxetine treatment that was 1,900 nanograms per litre. Now, the low and the medium fluoxetine concentrations in this particular study are reflective of levels that are found in the wild. And we also this time chose a high fluoxetine concentration that is above the levels that we might find in the environment, because these are the kinds of concentrations that toxicology studies typically use. So we wanted to have a baseline for comparison with more traditional toxicology studies. So let's go through the secondary stresses that we used in addition to fluoxetine. So Daphnia, in the case of the pathogen experiment, was subjected to one of 2 pathogen treatments, a control, which involved no pathogens, or a pathogen-exposed treatment, which involved exposing animals to spores of a bacterium called Pasturia ramosa when the Daphnia were 5 or 6 days old. Now, in this image here, you can actually see an uninfected individual up the top here and an infected individual down the bottom. And you can see some pretty notable differences. First of all, for the uninfected individual, you can see it's kind of opaque and parts of it are transparent. And in the transparent parts, you can actually see developing embryos inside the individual. But in the case of the infected individual, you can see that it's a different color due to the presence of spores. And there's also no offspring. So one of the effects of exposure to this pathogen is that it actually suppresses reproduction in Daphnia. And the other effect is that it actually can lead to gigantism. So the infected animals actually grow bigger. Okay, so here we have a cartoon showing you the general experimental setup. So the 2 different genotypes of Daphnia, M10 or HO2, that were exposed to the 4 different exposure treatments, and then each of these were either exposed to no pathogens or to spores of this bacterium. For a second set of experiments where we looked at temperature as a secondary stressor, the Daphnia were exposed to one of two temperature treatments, either 20 degrees or 25 degrees from day one of age. And here we have a cartoon showing this setup, very similar to the previous one, the two genotypes, the 4 different fluoxetine exposures, and then in this case, further split into either 20-degree treatment or 25-degree treatment. Okay, so turning to the results for this part of the research, does fluoxetine and pathogen exposure interact to impact life history traits? Well, we didn't find any effect on infection rate or the number of deaths, but we did find that fluoxetine, but not pathogen exposure, affected the age at first clutch. And in particular, high fluoxetine exposed Daphnia produced their first clutch more quickly than control and low fluoxetine individuals. We also found an interactive effect of fluoxetine and pathogen exposure on fecundity. So one of the general patterns to note from this graph is that infected individuals produced far fewer offspring over the 30-day experimental period compared to uninfected individuals, but this is not surprising. As I mentioned, one of the known effects of infection with this bacterium is suppressed reproductive output. But there are also some other things of note. In uninfected Daphnia, we found that low fluoxetine exposed animals actually had fewer offspring. However, we didn't find a difference in fecundity among the different fluoxetine exposed treatments in the infected Daphnia. We also found an interactive effect of fluoxetine, pathogen exposure, and host genotype on body size. So again, if you were to stand back and look at the general pattern, we find that the infected individuals, irrespective of fluoxetine treatment and genotype, were generally larger than the uninfected individuals. Again, this is not surprising because the other major side effect or impact of infection is gigantism, so the animal's expected to be larger. But if we were to look at the interactive effects, firstly, separating out the genotype, so looking only at uninfected HO2 Daphnia, we didn't find a difference among fluoxetine treatment. However, for the infected HO2 Daphnia, we found that individuals exposed to fluoxetine were significantly larger compared to the control. For the infected M10 Daphnia, we found that low fluoxetine exposed individuals here were significantly smaller compared to the other exposures. And for the infected M10 Daphnia, we found that individuals from the medium fluoxetine exposed treatment were smaller compared to the high fluoxetine exposed treatment. So some differences between the genotypes. Okay, so how does fluoxetine temperature interact to impact life history traits? So here we found no effect of fluoxetine on either the size of the first clutch or the age of the first clutch. But we did find an effect of temperature being much higher or stronger for the HO2 genotype than the M10 genotype. So indicative of a genotype by temperature interaction. We did find also an interactive effect of fluoxetine temperature and host genotype on fecundity. So again, we've split up the two genotypes and I'll go through the results in turn. At the lower temperature for the HO2 Daphnia, we found that those individuals exposed to higher temperature, indicated by the orange and the red dots, had higher fecundity compared to the control or the low fluoxetine exposed treatment. And at the lower temperature for the M10 Daphnia, we found that exposure to the low fluoxetine treatment in this instance had significantly lower fecundity compared to the others. However, at the high temperature, we didn't find any difference in fecundity among our fluoxetine exposures in either of the genotypes. We also found an interactive effect of fluoxetine and temperature on body size. And here at the lower temperature, for the M10 Daphnia, we found that individuals exposed to low fluoxetine were smaller than the other fluoxetine treatments. But again, at the higher temperature, we didn't find any difference in this case in fecundity in either of the genotypes. So a rather complicated set of results for the interactive effects of fluoxetine and other stresses. We found complex interactive effects between fluoxetine and secondary stresses on a raft of key life history traits. And importantly, we also found that genotype matters. Now, many of you sitting here watching the webinar would now be probably asking the question, well, how well do these studies, which have been carried out under controlled laboratory conditions, can translate to the real world? Now, this is a fair question, and is indeed one that we're now starting to tackle in collaboration with colleagues in Europe. So, for example, colleagues there have been able to expose fish, fit them with tracking devices and release them into the wild to monitor their fate under more naturalistic conditions. And I want to quickly show you a video that was lent to me by one of our collaborators. This is showing you fish that have been exposed to another antidepressant called oxazepam. So the blue dots are predatory pike, so you don't have to worry about that. What I want to draw your attention to are the yellow and the red dots. Those are European perch, which are pictured in the slide. The red dots are ones that have been exposed to oxazepam, and the yellow dots are the control fish, and you can see them swimming around in the lake after their introduction. And even without doing any formal statistical analysis, you'll see once the video speeds up that the fish that have been exposed to oxazepam, the red dot fish, are significantly more exploratory and active within the lake system itself. So using this kind of technology, we can hopefully monitor behavior and the fate of organisms under more naturalistic conditions. So I just want to finish off by thanking my various collaborators, particularly Michael Bertram and Lucinda Osselbrook, whose PhD research I spoke about in the second half of today's talk. And with that, I'm happy to take any questions. Thank you.
I have a number of questions, but actually, I'm going to allow the audience, if anybody has any questions, it'd be great if you type them into the Q&A, and I'll read them out, and Bob can answer them. Okay, we've got one already in the chat. I'll just remind everyone, if you could type it into the Q&A, that's fine. I can also answer it in the chat. So Raquel is asking, fluoxetine could not affect total number of offspring. However, what about morphology?
Yeah, that's a good question. I think in at least one of the studies, we did find a change in body condition of the fish as well. So this is something we've also found in with exposure to other pharmaceuticals. So I alluded to early in the talk that our lab group has also been looking at a veterinary pharmaceutical called trenbolone, which is used as a growth promotant in the livestock industry to basically improve meat yields in things like beef cattle. And in that case, it's an androgenic endocrine disruptor. We have found an increase in body condition even after short-term exposure in fish. So that's a good question. So I think exposure to there is evidence, both from our research on a couple of different pharmaceuticals and also from studies done in other labs that yeah, you can actually see differences in morphology as well. And I think it will be really interesting. This is not something we've done, but with something like an internally fertilized species like mosquito fish, I showed you the male's intromittent organ, the gonopodium. It'd be really cool, I think, to expose male fish from early in development and see if it actually affects the development of those kinds of structures, especially when we expose them to endocrine disruptors.
Great. And just along that same vein, just before I get to the Q&A, I had a question that follows along from there. And I'm wondering, in your first set of data, where you showed that the fish in Mexico, right?
Yes.
They had a decrease. There was a decrease in reproduction when they were exposed to the sewage water, but in Australia there was an increase. Did I get that right?
Yeah. So so in the case of the Mexican example, we did actually probe a bit deeper as to what in the water could be causing this problem. And as you saw from the picture of the Rio Canali, it's stained this very characteristic deep sort of tea color, which is associated with an increase in organic material. So in that case, we then decided to look at humic substances, which are actually naturally found in the environment, but we do get elevated levels with increase in nutrient input. And we found that, and we already know from previous research, the humic substances, because of the properties of the humic acid itself, has these hydrophobic properties. And researchers have shown that humic acid can actually bind onto the chemical receptors of fish. And they can also bind onto the sex pheromones of fish as well and render them undetectable. So through a whole series of experiments, we were able to show that humic substances actually impact the ability of the fish to smell properly, males, the odor cues of males of their own species versus males of the other species. It didn't actually change their motivation to mate. So when we presented females with the visual cues of males, they still showed very strong sexual interest in the males. But it does seem to be something with smell, because we also did experiments using food odor cues, and the fish weren't able to detect the food odor cues either when they were tested in water that was high in humic acid. Now, in the case of the mosquito fish back in Australia, we kind of thought that there might be some things in the water that could be interfering with the male's behaviors, and so we did target, screen for various substances, but none of the things that we screened for, which were the more obvious things like the contraceptive pill, seemed to be, could be responsible for the kinds of changes that we had actually seen. Like, we probably expected a complete opposite pattern if it was like an estrogenic endocrine disruptor, for example, that the males were responding to. So we weren't quite sure what it was in the water that was causing the shift in male behavior from the pristine site to the polluted site. But that was, I guess, among the very first studies that we'd done, and got us thinking about, well, let's now start looking at some of the more common pharmaceuticals that are turning up in waterways and just sort of hone in on impacts of those.
That's really interesting. I wonder, in terms of the difference between the two geographic areas, I think you mentioned that the species in Australia was invasive. And given the interaction you see between genotype and your later experiments, I wondered whether you can explain any of the potential diverging effects through the fact that some species are invasive and some are indigenous. Is that a possibility?
Yeah, that's a good question. I mean, I guess one of the things is that we tend to work on species that are easy to breed and oftentimes have become invasive species in many parts of the world. It would be really interesting, I think, to target perhaps a species that is a less successful invader, because you might expect to see some differences between species. The fact is, we're finding differences even among genotypes. Some of our research, which I didn't talk about today, we found differences in behavior in the response of males and females differing when exposed to similar concentrations of toxicants. So I think it's really important to focus at multiple levels of biological organization, right? So I think it's important to look at individual level responses. It's important to look at sex differences. It's important to look at perhaps population level responses within species, and also compare between species. A paper we published earlier this year showed that exposure to fluoxetine also erodes among individual variation. So animals end up behaving more similarly to one another. And if we think about variation as being really important in sort of mediating the response of populations, especially to environmental changes, then when we have a homogenization of, in this case, behavior, then we might actually, see populations respond less well when environments change.
Yeah, there's a statement in the Q&A thanking you for this really interesting but very scary data, and that I think that what you just described is just compounds the scariness, I think, of it all. So the question is, I wonder whether you've checked how long the effects persist, and if you've looked at subsequent generations, if there's any compounding effects as you go further along, I suppose, with every subsequent generation.
Yeah, that's another excellent question. So previous research has shown that exposure of organisms to certain pharmaceuticals like the contraceptive pill, even short-term exposure of adults can lead to effects that can transcend generations. So we know that exposure doesn't have to be long to have long-term consequences in subsequent generations. So in this case, exposure of mice had effects 2 generations removed from the actual exposure. In our lab group, we have been exposing fish called guppies, which are native to Trinidad and northeastern South America. It's a popular aquarium fish that's also become invasive in many parts of the world, including tropical parts of Australia. And we've been exposing guppies to Prozac now for several years, onto maybe our 15th or 16th generation now. The fish have been exposed to either fresh water or different concentrations of fluoxetine, and we've been repeatedly testing their behaviors over time. And what's interesting is that for some things, we're finding actually a persistence of differences that we saw even from shorter term exposures. But in other cases, there might be hints that long-term exposure could actually mollify the effects that we might see from shorter term exposure. And that might be because maybe the animals are getting used to or building up some sort of tolerance to those toxicants. So we have this massive experiment going on at the moment. We're still completing going through all the video watching, but we have been, these long-term guppy mesocosms, we've now also had done experiments where we brought the fish back into fresh water. So we want to sort of look to see, and then test their behavior over time. We want to see, yes, do we see this shift in behavior over time? And the longer they've been in fresh water, do we get perhaps a return back to sort of normal pre-exposure kind of behaviors? So yeah, really great questions.
Super interesting. I feel like guppies on Prozac could be a meme, but maybe you could maybe sell one of those gifts online, or anyway. So that leads quite nicely to John Bertram's question, which is with all the chemicals in the environment, what chance do we have of removing them in treatment plants? And as you just said, what does removing them actually do in the long term?
All right, so first of all, hi, John. I can't see you, but hello. Yeah, that's a really good question. I think that question needs to be tackled at multiple levels. A common question I get asked is, what can we do, as consumers? And I guess my first response is, please don't stop taking your medicines. But what you can do is maybe stop flushing them down the toilet or even throwing them in the bin, because there's evidence to show that these pharmaceuticals can even make their way into the environment as a result of landfill. And certainly here in Australia, there are programs in place where you can actually bring your medicines in to actually get back to the pharmacy to actually be disposed of in a way that is less likely to result in them entering our aquatic systems. Then I think another issue is that we need to educate our regulators. I gave a talk at a water conference a few years ago here in Victoria, in our state, which brought together water managers from across the state. And I was kind of surprised that the idea of pharmaceuticals in making their way through sewage treatment into the waterways was a bit of a revelation to them. And I think a lot of the focus has been on, I guess, things like heavy metals, and there's a lot of obsession over microplastics and other things at the moment. And so I think we really need to bring pharmaceutical pollutants onto the radar of our regulators. And by doing so, then hopefully we can also get the will to basically look at ways that we can treat our sewage better. I think some parts of the world are better at this. Scandinavia is a good example. They're already exploring ways of sewage treatment processing that could try and hopefully remove more of these toxicants from the water before they're discharged into the environment.
Yeah, that's super. I think that's an important point to make, especially in a talk like this, where you're presenting scenarios that are as we heard, pretty scary, and at least to hear that we as individuals can make at least some small token change in terms of being able to take pharmaceuticals somewhere to get processed properly. I don't know of a place actually in Canada. I'm gonna probably look that up, or at least around where I live. So just as we wrap up, I just wanted to bring this back to the worst type of animal, which is humans on our planet. And I found it really interesting, and I'd like to just know your thoughts on this about the interactive effects that you showed at the very end. And to me, it just screamed, social determinants of health in our population, and about how multiple things in our environment are going to impact on how we react and continue to react in that environment. So I have just a couple of questions there. So in that case where you show the interactive effects and perhaps changing reproduction and fecundity, whether you've looked at what happens if you allow the organism to continue to breed. Do they outbreed themselves in a do they end up being one of those invasive species? Do they just consume their environment and then die off, or do they adapt? Is there like a survival of the fittest where you get a kind of a subset that seem to be resilient, I guess?
That's a great question. So for the Daphnia experiments, unfortunately, we stopped the experiments at day 30. We would have liked to have basically track the animals for their entire lives. But unfortunately, because of COVID, and Melbourne actually became the epicenter of Australia's second wave, and our city was shut down for 112 days during the worst of it. So unfortunately, we had to bring that experiment to an end. But I think it would have been really cool to look at those kind of population level effects. We recently completed a study looking at community level effects, where we put fish into large pools with various other creepy crawlies that they would normally co-occur with in their pond environment, and then exposing the whole ecological community to things like Prozac, and then look at the relative abundance of the critters after the exposure, like sort of comparing before and after. So we're still analyzing that data, but I think you raised some really good points there. Can exposure to these things lead to population collapse? Can it alter community structure? Because animals don't exist in a vacuum, humans don't exist in a vacuum, and so I think these are really valid questions, and they're definitely things that we are interested in, and we both ourselves and our collaborators have been tackling.
And then just to wrap up, because your answers segue really nicely into my questions. The final thing is, animals and humans interact in the environment, and I wondered if you could speak at all to whether there's any information or data on Indigenous populations that still are consuming country food. So, for example, some of these species, I'm assuming, could be country food, and what happens in the environment when the food chain is tainted, and it leads it all the way up to Indigenous communities. If you can.
Yeah, I'm not aware in terms of the pharmaceutical work, but sort of thinking about a colleague at the University of Queensland has been studying heavy metals, Indigenous communities living adjacent to mining sites. And we see that these, manganese, I think it was, is showing up in the tissues of marsupials, these predatory critters called quolls on some of these islands. And there's also Indigenous communities right next door as well. So certainly these things do make their way in the environment and can obviously be taken up directly by the humans, but also, of course, through the food. And we know that in quolls, exposure to things like manganese can affect cognitive capacity. So the researcher, the colleague at University of Queensland, has designed some very simple cognitive tests on iPads that he's now testing children in some of these communities to study these effects. I guess the frightening thing is that we are seeing these toxicants ending up all over the world, even in really remote areas. I talked about pharmaceuticals in Antarctica. On the other end of the world, the chemicals are showing up in the tissues of polar bears as well. So I'm almost certain that these things are making their way through the food chain and including ending up in humans as well, unfortunately.
Yeah. All right, and on that note, I think we'll wrap this up. And thank you again for being so flexible this morning. We had lots of technical difficulties that we'll iron out in future webinars. But what a great talk! Really enjoyed it, and thank you for bringing this to the Dohat Society, where we don't talk a lot about ecology and life history theory in animals. So thanks so much, Bob. I know it's late for you and Bev there, and thanks to the ANZ Society for putting forward Dr. Wong's name for this great seminar. So thanks again.
Yep,
really enjoyed
pleasure.
it.
Thank you.
And thanks for
thanks.
everyone for attending.
It was great.
Thank you.
Great. Thanks, everyone. Have a good day, night,
night.
afternoon.
Thank you. Bye.
Take care.
Thanks.